Greetings and welcome to the Helix BioPharma webinar. It is now my pleasure to introduce your host, Heman Chow. And thank you, Mr. Chow. You may begin.
Thank you, organizer, and thank you, SNN, for allowing me this opportunity to share with you the story about Helix. There will be forward-looking statements that being made, and here's just a slide of the cautionary note that we have in place. Helix is a Canadian company. Our head office is situated in Richmond Hill, which is just north of Toronto, Ontario, Canada. We develop a unique platform of drugs. The platform is called DOS47 or DOS47. It targets the tumor microenvironment. Helix is listed on the Toronto Stock Exchange. We have about 133 million shares outstanding, 65 million warrant units outstanding, and our market cap is about 70 million Canadian dollars. We are followed by Noble Capital and SEDAR Small Cap Research, and the target range there is about $2 to $2.50. We currently have cash at about $3.3 million, a very clean cap structure. We have no debt. Helix management, board of directors, and scientific advisors are very capable individuals. I'm very grateful to be working with these leaders. They span experience from running large corporations to finance to expertise in medicines and scientific research. And particularly, I want to highlight the wonderful relationship company is able to have with Moffitt Cancer Center, specifically with Dr. Robert Gillies, who sits on our scientific board. Dr. Gillies is a world-renowned expert in imaging, in the tumor microenvironment. And in fact, if you look him up in scientific literature, you'll notice that he's the leader in tumor microenvironment, specifically on tumor metabolism that relate to tumor acidity, which we know now that suppresses the immune system. Lung cancer and pancreatic cancer. These two are the diseases that no doubt most of us know are a big burden to the society. In the American Cancer Society publication for the estimate in 2020, lung cancer is going to be the #2 cancer for both men and women. The total number of cases they expect exceed 200,000 cases. Pancreas, in both men and women, cases will exceed 57,000 cases. And in 2020, the estimates for death, unfortunately, in lung cancer is 135,000 and above. And pancreas is really terrible, 47,000 with a 5-year survival rate percentage of well below 10%. These are the two diseases that Helix is trying to make an impact on, and these are the two indications that we developed, our lead drug is targeting. But before I go on to explain about our drug, I just want to give 30 seconds about the way that we target tumor that is very different from everybody else. We learned a lot in the past few decades about there are genes in cancer cells that are different from normal cell, and some of these mutations will drive the growth of cancer development. And then the last 5 years or so, we suddenly realized that while it's not only about the gene, isn't it? It's really about the whole complex system. The tumor really is almost like a living organism on its own. And more importantly, we realized that the tumor cells, in fact, can communicate with the immune system around it, giving signals to tell the immune system that, no, you need to shut down, do not attack us. Success with these checkpoint inhibitors, so-called Keytruda, for instance, nivolumab, really open up a brand new avenue of how we can treat cancer, realizing that if somehow you can disrupt this tumor suppressing effect of the immune system by the cancer cell, you can open up a brand new way of dealing with cancer. And the wonderful success of Keytruda, for instance, in lung cancer, melanoma, and certain other cancers are really giving us a whole new way of thinking about cancer development. Helix contention, however, is the following. The complex tumor microenvironment and the crosstalk between the cancer cell and the immune system is not just a simple dialogue. In fact, it's been known for many years that cancer cells have a very specific way of utilizing energy. And in their own metabolic profile, it generates a whole host of metabolites. One of those is lactate. It's been known for years, literally for 40 and 50 years, that lactate can be found in abundance in many, many solid tumors. But people have always thought this is just a waste product, a necessarily result of biochemistry of the cancer cell, and not much is given thought to it. However, you and I both know when we do a lot of exercise, our muscle ache, and that's because of the accumulation of the lactate. And you and I intuitively know what, there's got to be a little bit more than just this is a waste product. Well, we know now, especially in the last 5 to 10 years, this lactate, in fact, is one of the first reasons giving a signal to suppress the immune system. And the way by doing it is when you have accumulation of lactate, you actually lower the surrounding pH. So this tumor acidity issue that's been talked about in scientific literature, been talked about it in popular literature, but people really kind of just looked at it as a curiosity. People have pointed out that if you really don't control tumor acidity, you might not really fully realize potential of chemotherapy, for instance, and more recently about immunotherapy. However, no one has yet to really develop a therapeutic that you can obtain in the market to say, well, can I suppress tumor acidity? That drug is not available yet for cancer. What Helix has done is put forth an idea that we can combat this tumor acidity, and in fact, the platform has enabled us to develop a drug called LDOS47, and this drug is given to patients by IV, and we have taken this drug in multiple clinical trials, first in non-small cell lung cancer, and now this year we're entering pancreatic cancer. So LDOS47 or LDOS47 is an antibody enzyme conjugate that we give to the patient through IV, and its purpose is to get to the solid tumor. It sits on the outside of the solid tumor, and it uses the body itself, urea, a waste product, and convert that metabolically to ammonia to generate this ammonia high pH environment to directly counter the acidity of the tumor. That is how our drug works. LDOS47 is in a number of clinical studies. In front of you, you see the light color boxes, the light blue boxes. They represent LDOS47 in different clinical trials for non-small cell lung cancer. The top line represents monotherapy. These are lung cancer patients that have experienced, unfortunately, as many options that the doctors are able to give it to them, and they came onto our study for safety and potential efficacy studies. And we completed the phase 1 for monotherapy. We've entered partially the phase 2, and we have some data on the phase 2 side, but then we decided to close out the study and move into the following boxes. The reason for that is because in cancer development, we realize and we understand that it is in the combination approach that you're going to give the best benefit for patients. And therefore, for LDOS47 development, we have moved into combination trial. And in the 2 boxes below it are 2 types of combinations on 2 different types of chemos. The second box line there is combination with 2 drugs that are used exclusively almost in certain part of Europe, in certain part of Asia. The third line box there is LDOS47 with pemetrexed carboplatin, 2 chemos that are used commonly in North America and in Western Europe. And the last or the 4th blue box there is something that we're doing now in the preclinical work, especially with Moffitt, where we are testing LDOS47 with immunotherapy, something that we want to get into clinic when all the conditions are met. The green box represents pancreatic studies, and we have just initiated that at the beginning of this year, the end of last year. We have enrolled patients already. This is a phase 1b/2 study, we will enroll 9 patients in the phase 1B and then 11 patients for phase 2 of the study. And I'll mention more of that as the slide goes along. Can you see tumor acidity? And yes, you can. On this slide, on the top panel, you will see 3 black boxes. What you're seeing there are actually MRI pictures. These are MRI of a mouse that has a pancreatic tumor. The pancreatic tumor is on the lower left-hand side, that round green outline that we show on the bottom left. The first picture is just a typical MRI picture showing the structure of the tumor. The center picture is a very special imaging technique that we apply to this MRI, and this is being done in Moffitt Cancer Center. And what you're seeing there is you can actually ask, what is the pH of this tumor? And in this particular example, the pH of this tumor is well below 6.5, closer to 6 of the pH. Neutral pH is 7, so anything below 7 is considered to be acidic. The picture on the right, the last picture, is what happens to this tumor when this animal is given our drug 30 minutes later and we take a snapshot. You can see the tumor begin to change from the acidic, moving back towards the neutral. And you can do more sophisticated mathematics and laboratory experiments, and that is shown on the bottom left. I won't go through all the details, but suffice to say, if you compare the blue and the red, the blue is what happened before you inject our drug. The red is what happened what happens after you're injecting our drug. The difference between the two, you can do the math, and it tells you that in that duration of time, you shift the pH for at least half a unit, perhaps even more than that. So tumor pH is acidic. In these examples, our drug can change pH. Does it really matter at the end? And what I'm trying to show you then is an experiment on the bottom right. This case work done by Moffitt Cancer Center using this new mouse model that we have developed for pancreatic cancer. The black line showing the growth of the tumor on its own, and the red line is what happened when you give our drug together with a drug that we know will help to release the immune system. The data is very dramatic, but this is still the first time go around, and Moffitt is looking to repeat this and further delineate the exact relationship between our drug and the immune system and the pH. But this is the kind of data that encourage us to realize that this idea that if you don't work with the tumor acidity issue, you're really not seeing the full potential of immunotherapy. This is the kind of data set that encourage us to move forward in that direction. Let me quickly then bring your attention to some of the clinical data set that we have already. What's in front of you is data set about our monotherapy, that is our drug alone, in advanced metastatic lung cancer patient. We have dosed patient in groups of 3. A cohort is a group of 3. We have dosed all the way for 16 cohorts. And what's in front of you on the bottom chart is seeing what happens to the tumor of this patient in the last 7 cohorts. What you see here, and particularly in the red, are those patients where the growth of the tumor is controlled. So we call that stable disease. The red represent those stable disease that are longer than 16 weeks. And in this scenario, as we're told by the doctors, that any patient that can control the tumor by our drug or by any drug over 16 weeks is a meaningful result that we should further explore. And it is this kind of a data set that encourages us early on to explore more about this in combination with chemo and so on. More recently, and that is about 2 months ago, in the ASCO, the Association of Clinical Oncologists in the United States, we provided the first glimpse of our data, our drug, together with 2 chemos, pemetrexed and carboplatin. This is a study done in the United States. In this particular studies, we were able to enroll 14 patients. Out of the 14, we were able to examine the data set for 12. And out of the 12 patients, we were able to see 5 of them that have a tumor response that is classified as so-called partial response, which means the tumor shrank by more than 30%. And then we also have 4 patients that have stable disease. And then if you do the math, the doctor tells us that this means an overall response rate of 42% and then a potential clinical benefit rate of 75%. This is very encouraging to us. It tells us that our observation with the monotherapy is continued to be upheld in this kind of studies. It tells us that Aldose 47 in combination with these 2 chemos are safe. Potentially, in fact, it may be efficacious, but that is too early to say. This is only 12 patients here, but it is this study, the monotherapy studies, and some of the preclinical data set that is telling us that when we move forward in non-small cell lung cancer, we have the potential of chemo combo and perhaps the potential with immuno combo. Let me quickly switch gear and tell you some more about our pancreatic study. This will be the last slide on our clinical data set. In this particular study, we are treating patients that have gone through at least 2 rounds of medicine already. And so these patients come to our trial, unfortunately, really do not have too many choices. These patients will be given our drug together with a chemo. And this is not just any chemo. This chemo that we have shown preclinically, which is the bottom slide here, that if you're able to manipulate pH of the environment where this chemo is, you may improve the activities of this particular chemo. The bottom left show you what happens when this drug is given with our drug and the pH change in a lab bench experiment. The red show you the dramatic difference if that compared to the black line, which the chemo was given alone. The graph on the right show you, in this case in animal studies, that if you give our drug first and then the chemo with a 24-hour benefit, you can definitely see a significant improvement in how you can treat tumor. And these are the preclinical studies that have helped us in supplying information to the FDA that enable this particular pancreatic studies. Some of the key milestones for this study is by the end of this year, we're looking for the phase 1B portion of the study enrollment to be complete. And then we're hoping that by the end of next year, the phase 2 portion of the study, the enrollment will be completed. However, we are, as every company is, facing COVID-19 restrictions, whereas the hospital really need to diverse resources in ready for this pandemic. We do have our own hurdles in terms of enrollment, but we have made a mitigation plan, and part of that plan including opening sites. So some of the projections we're giving now is assuming that we're able to open new sites and therefore keep up with our enrollment with minimizing any delay. The next slide will show you now a little bit of the milestone picture that I've just described. So in 2020, the U.S. phase 1 in non-small cell lung cancer, the data set we have disclosed, and that was an ASCO that I have just mentioned. In the pancreatic side, I mentioned that the phase 1B, we're looking for enrollment complete by the end of 2020. And then as we move forward to 2021, we hope that the pancreatic cancer phase 2 portion in terms of enrollment will be complete by the end of 2021. And then we're actually hoping that if the preclinical data set continue to be positive, that we'll be in a position to look into a phase 2 immunotherapy combo studies in lung cancer. And then in 2022 and 2023, we'll see these studies come to completion and maturation in the pancreatic side. In fact, we're hoping for success at the end of 2022 and end of 2021, allowing us perhaps entering into a pivotal study in 2022. But there will be a lot of conditions that we need to meet, both in terms of clinical data, in terms of finances, and of other corporate conditions that we need to meet. However, we're hopeful, and these are the programs that we are pursuing. Now, overlaying all of that, of course, is work that we're doing now in terms of making the company financially sound and supporting all these milestones. We are in the process of raising funds. We're looking to raise $15 million USD, At the same time, we're hoping that raise will enable us to list on a US stock exchange. We are listed on the Toronto Stock Exchange, so that gives us some benefit of transparency when we do this kind of a cross-border deal. The time frame of this is somewhere between 3 to 6 months. And again, both COVID-19, the difficulty is in communication, and of course some of the restraints with corporate listing on NASDAQ, for instance. We still need to work all of that out, but we are well in our path in working with the necessary parties to enable us to do this raise. One thing that I have not mentioned, and that is we actually have a European subsidiary, but this year we are in the process of divesting it. This subsidiary and actually have licensed from us all the assets we have on cell therapy. They're going to go on to become a fully fledged company developing cell therapy. So we maintain a licensee relationship with them. But in the course of this process, we're divesting the rest. We have signed a term sheet that by the end of August, the rest of this European subsidiary will be sold, and we're looking of gathering at about $1.7 to $1.8 million USD to complete that deal. So here comes to my final slide. Just to quickly summarize, Helix developed a novel anti-cancer platform. We're dealing with tumor microenvironment, specifically about lactate and tumor acidity. We believe we are the leader in this because we are the one that would a drug that specifically addresses this in multiple clinical studies. We have some very interesting milestones coming up in latter half of 2020 and 2021. We are active in the market now, looking at fundraise, and if everything works out nicely, and I hope so, that will be cross-listed onto the NASDAQ, targeting the second half of 2020. So I'm going to end my presentation there, and I just want to thank you, all of you, to participate and listening into this particular presentation. So if there are any questions, I'll be happy to address. Okay, at this time, there are no other questions. So in that case, I will end my presentation here. However, if there's any follow-up questions that I did not make clear, or there's some additional information that you wish to have, you are more than welcome to contact me directly. My information that can be obtained from the conference organizer. I apologize, there might be some question here. Let me just double check on the question deck here. There was a question here by a question about any chance of a partnership with Big Pharma. The answer is we're definitely working on that. So we are presently engaged with Deloitte, the Canadian division. They represent us in reaching out to biotechs and Big Pharma, and we are in dialogues. However, at this present time, we have not disclosed any particular deal, but Helix, our business and operating objective is the following. We'd like to develop our drugs to a certain key data set and allow the Big Pharma to take it away to do the full-fledged large program. At the same time, because we're looking at combination studies, we are actually active talking to people about their drugs, and then whether, in fact, by combining with them will make sense. So the simple answer is yes, and we are using Deloitte Canada to help us to make those discussions. Okay. I don't see any more questions coming in. In that case, I'm going to close off the presentation. I want to thank you for joining in and listening to the story about Helix. Organizer, I'll pass the time back to you. Thank you.
This concludes today's webcast. You may disconnect at this time. Thank you for your participation.